Hi, my name is Lima-Melo, and here I'm going to present the consequences of photosystem 1 for inhibition, lessons from the Arabidopsis pgr5 mutant, as a participation in the first Brazilian Symposium on photosynthesis. As you know, photosynthesis is one of the most important biochemical processes on Earth and can be divided into two main steps. In the first phase, photosystems 1 and 2 use energy from light to drive electrons from water through the transport chain to the second phase of photosynthesis. In the second phase, the electrons provide reducing power to drive the CPP cycle to assimilate CO2 into more complex molecules, such as carbohydrates. When electrons accumulate in the electron transport chain, usually has a consequence of low electron sink capacity or excessive electron pressure in the chain, as occur under highlight conditions, for example. A condition named photoinhibition may occur. However, most of our knowledge of photoinhibition concentrates on photosystem 2 photoinhibition, while only recently we are starting to understand photosystem 1 photoinhibition. Therefore, in this work, we try to answer a simple question. What are the consequences of photosystem 1 photoinhibition for plant metabolism? So, trying to answer this question, we used an approach of combining a condition of excessive electron supply, which was the highlight treatment, with an Arabidopsis mutant leaking a photoprotective mechanism against photosystem 1 photoinhibition, which was the proton gradient regulation 5 mutant, the pgr5 mutant. We then confirmed and discussed the photosystem 1 photoinhibition under these conditions, and the following photosystem 1 recovery was also investigated, and we used different processes for to investigate plant metabolism. The plant metabolism, such as techniques of biochemistry, biophysics, molecular biology, and plant physiology in general. As an overview of our results, we showed that the use of pgr5 mutants treated with highlight is a good model to study photosystem 1 photoinhibition and its recovery. This occurs because this mutant displays a low thylakoid membrane pH gradient and thus lower cytochrome B6F regulation of electron flow, which specifically induces photosystem 1 photoinhibition under conditions of electron pressure at photosystem 1 donor site, such as highlight. So our results show that photosystem 1 photoinhibition, as expected, decreases the CO2 assimilation rate, and as a consequence decreases also the carbohydrate accumulation in mitochondrial respiration. Surprisingly, our results also indicated that photosystem 1 photoinhibited plants accumulated less reactive oxygen species, which results in lower levels of oxidative stress. However, the lower ROS accumulation also resulted in lower lipid oxidation and its consequent oxylipine accumulation. Because oxylipines are important signaling molecules, photosystem 1 photoinhibited plants displayed misregulated chloroplast retrograde signaling. As the main conclusions and insights of our work show that photosystem 1 photoinhibition is rapidly induced under conditions of reduction pressure imbalance between photosystem 1 donor and acceptor sites, which severely inhibits CO2 fixation, carbohydrate accumulation, and mitochondrial respiration. Plants are able to rapidly recover the CO2 fixation despite photosystem 1 inhibition by improving photosystem 1 efficiency through the light harvesting complex 2 phosphorylation and activation of reserve photosystem 1. The chloroplast regulation of nuclear gene expression is dependent on photosystem 1 activity under highlight stress through enzymatic oxylipine synthesis and hydrogen peroxide production, and that the inactivation of photosystem 1 can be a protective mechanism against oxidative stress in the chloroplast stroma and in the wider cell by preventing ROS overproduction. So the full results can be found in these 4 publications and also the respective discussions. And you can also contact me through this email. And finally, I thank the financial agencies, the universities, and all the co-authors involved in this work. Thank you.